Welcome you to the 35th CCMB Foundation Day celebrations. I'm Aishwarya, a neurobiology faculty, and I'll be taking you through the events of the day. We're absolutely privileged to have with us today Professor Ajay Sood, the Principal Scientific Advisor to the Government of India as our Chief Dignitary. Thank you for joining us, sir. Your presence definitely makes this occasion grander. We have a blend of stimulating talks and celebrations lined up. And to get us started, I now welcome our director, Dr. Vinay Kumar Nandikuri, to deliver the inaugural address all the way from London. We thank him for addressing us even while being away on an unavoidable personal trip, hence the virtual mode. Vinay, the screen is all yours.
Thank you, Aishwarya. At the outset, I would like to apologize for not being there in person. Due to a family emergency, I'm away. I thank the Principal Scientific Advisor, Professor Ajay Sood, for accepting our invitation to deliver the Foundation Day lecture. I would also like to thank all the dignitaries, though I can't see anybody on my screen, colleagues and scientists and students who are there. Today, I would like to start the report with movements from the CCMB's past. Our founder director, Dr. Pushpa Mitra Bhargava, envisioned an independent institute focused on frontier areas of biology all the way from 1960s. It took a lot of effort from him with the support of then RRL director, Dr. Sidhu, and then DG CSIR, Dr. Naidamma, to materialize this vision in 1977. At the initiation of DG CSIR, a high-powered committee consisting of Obek Siddiqui, Prant Talwar, S. Ramchandran, TV Subbaya, and Pushpa Bhargava met to work out the possibilities of setting CCMB with an initial budget of 3 crores for the first 5 years. This was approved at multiple levels, and finally, on 1st April 1977, CCMB was formally set up with Dr. Pushpa Bhargava as the founder director. At that time, it was set up in RRL, which is what we now call as ISCT, and it had a space within RRL. And the first annual report that came out in 1978 is essentially Dr. Bhargava's group work. In 1979, the report that I was looking at had 5 papers listed, and 2 of them are back-to-back Nature papers. With this, I would like to go to the presentation that I had prepared for this today. I hope everybody can see the presentation. I hope everybody can see the presentation. So the inception and inauguration of CCMB, I gave you a background about. It came about in 1977, 1st April. The building that we all are housed came up in 1987, and it was inaugurated by then late Prime Minister Shri Rajiv Gandhi. And interesting is there were 7 days of symposiums that were organized by founder director Pushpa Mitra Bhargava, out of which one of the days was a symposium on arts and science. You can see this is a picture of late Prime Minister Rajiv Gandhi, Shri Rajiv Gandhi, giving a lecture at the inauguration. And here are some of the other pictures by Dr. Francis Crick and the Nobel laureates, Dr. Ochoa. These are not the only ones, but many people came from across the world for this inauguration event. So, and this inauguration event also had performances by I'm still having problems with my screen. Okay. Bharat Ratna Ustad Bismillah Khan, Padma Shri Shobha Naidu, and many other events. And what you see here is the Hussein, the wall that we keep on looking at it. And it was actually done by Padma Vibhushan, M.F. Hussein. And this is one of the images that was given to us, given to me by the team. So this is the honor board of 1987 CCMB. There are two faculty here, M.R. Das and Balasubramanian, and the rest of them are basically, some of them are from CSIR headquarters, some of them from CCMB, but this is the total team at that point of time. And these are the photographs from 1987 who went on to become the directors of CCMB, Sharat Chandra, D. Balasubramanian, and Lalji Singh, who are our second, third, and fourth directors of CCMB, post Pushpa Bhargava. And one of the things about CCMB it has an amazing culture of having artists coming here and performing these workshops. And this is one of the images from 1999 artist gathering. And that's why you see a lot of pictures and a lot of paintings in CCMB, which is unique. So this is the background. With this, I want to talk about CCMB today and basically talk about our work today. So at that point of time, CCMB had one campus, that campus that we all see. That was 1987. At this point of time, we have three campuses. Of course, the main campus is still the same. And NXC is approximately, around a 5-acre campus called what we call as NXC, where we have Atal Incubation Center. In addition to that, we have Lacones, which is located around 20 kilometers from the main campus, which is around 7 acres. And this is where the conservation work happens. We are now 950 people. Compare that with the small honor board that I showed, with 148 PhD students, 46 postdoctoral fellows, and 39 project leaders, and many other people involved in support and other activities. In the past 1 year, we have had many new colleagues joining CCMB. Professor Vasant Shinde, who has come here as Bhatnagar Fellow, is a former Vice Chancellor of Deccan College and founding Director General of National Maritime Heritage. Janesh Kumar, who's basically a senior faculty at NCCS Pune, joined CCMB very recently. Same is the case with Santosh Chauhan, who used to be at ILS, joined CCMB very recently. In addition to that, some young colleagues like Dhananjay Titarvidi and Sriram Varhan joined CCMB in the past 1 year. These are the other new colleagues who have joined and will be taking care of, in addition to being scientists, they will also be taking care of the facilities. Mega will be taking care of the zebrafish facility. She's joined us in April or May. Shiv Prasad Singh Yadav, Ravi Kumar Reddy, all of these people from various places joined as a scientist at different levels in CCMB. We've been pretty fortunate to our scientists are recognized across the everywhere, and the major one is Dr. Shankar Narayan is now an associate member of EMBO. So the way it goes is that EMBO members, when they are EMBO fellows, when they are from there, like within European Union, they are called fellows, and when they're outside the European Union, they are called associate members. It's a major privilege, and it's not an easy thing to be. And congratulations due to Shankar Narayan for becoming the associate member of EMBO. And in addition to that, Giriraj Chandek got the 5th Venus International Healthcare Award, and Umapati for Gold Medal Oration Award, and Sachin Singh for oral presentation at an international conference. These are the few awards that our faculty colleagues got. In addition to that, our students have been doing exceedingly well, and they have been awarded Best Poster Awards in various places. For example, Pradeep in HiSci, Vinod in SRBCE conference, Anusha and Hanuman Kale, all of them have been awarded Best Poster Awards in SRBCE conference that was organized recently. These are in addition to that, India Cell Biology Conference, Raj Bahadur, Best Poster Award for HiSci in Annapurna, and India Cell Biology, once again, the award for Devaprata Jana, Amareshwar. There are many awards. These are the Best Poster Awards and other things. In addition to that, we also have Sakshi getting a Best Poster Award at seminars on crystallography, Prakash getting in third place in INEAS, Saransh, Bharati, and then Gayatri, Rohitesh, all of them got awards at different places across India. And we also got many travel awards for students to visit various conferences.
Sir, you are muted, sir. Excuse me. Sir, excuse me, sir. You are muted, sir.
Now, is it okay?
Yes, sir. Yes, sir. Sorry, sir.
Which slide onwards I'm muted? Just now. Okay.
Before sir.
So we have many major projects. We collaborated with across the thing with INSACOG, and we actually participated pretty heavily in COVID sequencing. SBI Foundation gave us approximately 10 crores, and this was also for setting up satellite centers and other things. Indian Breast Cancer Genome, Welcome DBT Intermediate. We have 5 fellows with us, Genome India, and other agri-nutrient and other things. In addition to this, we also have many other ongoing projects, and this is something that CCMB has pretty much done very well in participating with multiple agencies to write major projects. I would not go into the details of publications, and generally I do that in the Research Council meetings. We have published 152 papers from last November till now, and these include papers in eLife, PNS, EMBO, and Nature, Science Advances, PLOS Genetics, and many other journals. These are few examples that I have put up here for people to appreciate the kind of work that is getting done. We have a very strong business development unit headed by Archana Siva, and where we do many things, including contract R&D, molecular services, analytical patents, agreements, and other things. In the past 1 year, approximately 3 technologies have been transferred, and we have many patents that have been granted, MOUs that are I'm still trying to figure out my screen part of it. Okay, patents filed, granted, all these things have been done. In addition to that, the business development unit is involved in a lot of training activities, including SDP, summer training, PBST, and we have graduated 22 PhDs in the last 1 year. So science communication is something that we are very proud of and very strong at. This is headed by a lot of work is done by Somdatta and Ramakrishna for this. We have blogs which talk about science, CCMB science, which are scientific tales and zines, dark, and many videos that we generate, and these are widely seen by people. And in addition to that, we have many one-day events. These events basically, including an open day where this time we had around 8,000 visitors now across Hyderabad, coming here on 26th September to CCMB. And we also have a wildlife week celebrations. We have public lectures. One of the things was given by our colleague and CDFD director, Tangaraj. And we have study tours conducted for students so that they can come and have a look at what kind of equipment, what kind of things happen in CCMB. In addition to that, we have week-long programs at CCMB, such as Young Innovators Program, and many other small programs wherein people come, selected students come here and spend time in CCMB. I interacted with these students, and the enthusiasm is palpable, and these students are stimulated after being there and looking at what kind of work gets done, and hopefully some of them will come back and take up science in a big way because of these visits. We have had some interesting events and important events at CCMB. Of course, there are two conferences that were organized at CCMB in the last 1 year. One of them is on omics, and the other one is on reproductive biology and comparative endocrinology and development. These are had at 54 invited speakers and lots of participants. It was an amazing event. It was pretty much widely seen, widely attended by the people and appreciated by that. We had Founders Day on 22nd February, which we started around 3 years ago after when our founder director, Dr. Bhargava, passed away. And this time the lecture was given by then Principal Scientific Advisor, our ex-Principal Scientific Advisor, Vijay Raghavanji. This was an event where a lot of ex-CCMB alumni come and interact with the current students, and it's an interesting event, and important one. We also had a visit by Colombian delegates, which you can see here. And in addition to that, we actually have many visits of by many people across. I'm not talking about everything. Atal Incubation Center has completed 5 years. So Atal Incubation Center is essentially our Section 8 company, and it started in 2017, and it has completed its 5 years tenure now. So this was formed by CSIR CCMB in 2017 with the support of Niti Aayog, and it is called Atal Incubation Center, and which is basically has been recognized in the top 10 in the Biospectrum magazine for last 3 years. We have been in the ranked top 5 in the biotech incubators in India. What's interesting and important is that we have had many companies that went through AIC CCMB, such as, basically that is how we are actually helping the community in a big way. There are many programs, physical and virtual, BIRAC, supported by many people, and you can see some of them here. What's important is what here is dashboard. We have 85 plus startup companies that have been supported in the last 5 years. 27 IPs have been generated. 23 awards and innovations have come for these incubatees. We have invested 5 crores in various other things, and current number of incubatees are I can't see in the screen. No, 28, and there are 2 new branches, one in IGAB and one in Imtech, that have been opened. It's not they are not operational yet. So in recently, AIC CCMB and CCMB organized a lecture series on microphysiological systems and advances, an EMBO lecture series that was organized for 4 days. And we have also, as I said, celebrated our 5 years of AIC pretty much very recently. And this is the team of AIC, which is headed by Madhusudan Rao, but our chief operating officer is Ramji. And you can see the other people, including Ritika. These are all the team that is involved in AIC. I would like to close it with what we actually have done. I know the pandemic is now kind of going to become an endemic, and it's kind of gone down a lot. But last 2.5 years have been pretty much where a lot of work has been done on COVID by CCMB. We worked on diagnostics, we worked on vaccines, we worked on some of the treatment testings, and we have done a big way on the surveillance and on COVID mitigation. We have given a lot of seminars. Previously, this was done by various people, including Rakesh at a later point of by me. We give interviews in the media to make sure that people are aware of what kind of what is happening with respect to COVID. We have sequenced approximately 40,000 samples in the past 2.5 years. Actually, it's not even past 2.5, it's around 1.5 years since INSACOG has been set up. And we were tracking the lineages, and we continuously track them and look at the emergence of newer lineages. This is about the Omicron and Omicron sublineages. And one of the other major things that has happened in the last 1 year is the development of mRNA platform for vaccines and biologicals. This is important for the future preparedness. This was work done by Atal Incubation Center and CCMB and CCMB in collaboration. This is basically essentially what we have done is what we designed vectors that would express the spike protein. This is already a technology that was in place and completely based on the Moderna technology. But what we have done is we have gone from proof of concept from end to end, where we have actually cloned it and expressed it, purified it, generated a lipid nanoparticle, put it into hamster, performed a challenge experiment to see that the whole process works. We are most probably first academic institute which is completely and independently done this work from end to end, prove the proof of concept. This has large implications. Now we know how to do it. It can be extended to many other diseases and many other aspects. Key challenges are production of high quality mRNA, and major step forward is freedom to operate in India. The work that is done at CCMB and this is the sewage surveillance work, which is done in collaboration with IICT, NIRI, and NCL also has done this work along with us. So this COVID sewage surveillance work was recognized by Golden Peacock Eco Innovation Award in 2022. This was awarded to CSIR for the work done at our institutes. This other thing that recently we were recognized is the dry swab technology for COVID-19 diagnostics. This received Technology Tech Transfer Impact Award recently. This was conferred by STEM Society. So, as I said, we've been pretty much CCMB is a well-known institute within in Hyderabad and around all over India. And so we've been in the news. We have we give press releases regularly about our scientific findings and many other things. And so with this, I would like to say thank you and I would look forward to the presentation by Professor Ajay Sood.
We thank our director for the excellent inaugural address, highlighting the history and genesis of CCMB, which took us down the memory lane, and for sharing a complete overview of all institutional activities from the past year. It is now time for the much-awaited Foundation Day lecture, and I invite my colleague, Dr. Sriram Varahan, up on the dais to formally introduce our speaker, Professor Sood.
It is my distinct honor and privilege to introduce the chief guest for our 35th Foundation Day, Padma Shri Professor Ajay Sood. So Professor Ajay Sood is currently the Principal Scientific Advisor to the Government of India. A physicist by training, Dr. Sood is one of the foremost authorities on hard condensed and soft condensed matter physics in the world. Professor Sood and his team created history in 2003 when they generated electricity by passing liquid or gas through carbon nanotubes. This discovery, referred to as the Sood effect, in honor of our principal scientific advisor, has numerous applications ranging from self-powered pacemakers all the way to gas pipelines. Professor Sood continues to push the boundaries and frontiers of applications of physics by incorporating these nanotubes in various materials, strengthening its structural integrity without adding weight to it. A little bit about our principal scientific advisor. Professor Sood received his PhD from the prestigious IISc in 1982, and he did a short postdoc at Max Planck, Stuttgart, Germany, after which he joined the faculty of his alma mater, IISc, in 1988. Where he had an illustrious and long career and served as the chairman of the Division of Physical and Mathematical Sciences till 2008. He has been decorated with various prestigious awards, including the highly reputed Shanti Swarup Bhatnagar Prize, the TIWAS Prize in Physics, as well as the J.D. Birla Science Award. We very much look forward to your talk titled Nature Inspired Science, and without further ado, we would like you to come on stage.
Yeah. So first, let me thank the people who have been contributing to this. So all this interest in this field started with my student Nitin, and then Shriram has been my constant companion in this field, because this field becomes very exciting when you combine theory and experiment. And he is one of the person who is a pioneer in this field. His students Rahul and Harsh, and my other students who have joined, Pradeep, he finished, and the ongoing student is Roshan. And very recently, Professor Ganpati from JNC, which is an institute in Bangalore, 10 kilometers from ISC, has started very exciting experiments in active matter, which I'll tell you in the end, and his student, Pragya Arora. So that will be the last part of my talk. Oh, it's not changing. It's not changing. Something I'm not doing right. No. Do I have to change here? So let me see whether it changes. No, it's not changing. Sorry, it's not changing. So I have to do probably the, maybe you can keep it here if you don't mind. Then I can. Yeah. Okay. So first I'll like to tell you what I will not talk today. So we are all familiar with this fascinating phenomena or observation called Brownian motion, where you see this, for example, dust, small particles of dust or colloidal particles of any shape and size. They perform the random Brownian motion. And this one, oh, sorry, it got. So it's going to be a challenge. Okay. So here, what is important is that it has a Brownian, like a drunkard moving on a platform. So the position of a particle does not go linear in time, which is what you will put velocity into time, but it does not go linear. It goes square root of time. So that is what we all know from our high school. What I'll be talking today is not Brownian one, but something which you see in your lab, that if you look at bacteria, for example, a lot of you are experts and work on it, E. coli, and look under a microscope, they'll be moving in a very directed manner, which is what is the trajectory being projected here. But very fascinating thing is, which is what attracted me to this field, that when, oh, okay, let me just do it here, doesn't matter. No, let me do it here. But when you put many of them together in a Petri dish, those bacteria perform highly coordinated motion. So you can see the group of them moving collectively. And this is something you also see when you go in a forest or a garden and you see these ants going around almost for infinite time. If you have a patience to watch, they can go left-handed, right-handed. That's not important. But this is what, if you Google ant mill, you will see beautiful photographs of this ant mill, and you can probably spot one in your garden. And these are the marching locusts, the locusts which put on a Petri dish. Above a certain number, they again perform highly coordinated and coherent motion. So this is all collective phenomena. And this is another animal which comes to our attention, that if you put these animals in this concerts, and if there is the right music, you will see this beautiful collective behavior. But please remember, this is taken from a very prestigious journal called Physical Review Letters. So there is a whole lot of physics behind this, and I will try to address some of that. So this phenomena, which you all know, English word flocking, is a collective motion of so-called living organisms, and I'll show you it is not only for living, it could be for any active matter, by sensing each other's presence, orientation, and direction of movement. And this is the emergent behavior. Emergent means individually they perform something, but when you put collectively, they performed behavior which is the emergent, which is not really having any central coordination. They all come together and perform this. And these are some of the examples, even for fishes and these birds, which you might have seen in the sky, having these thousands and lakhs of birds in the sky. And if you're lucky towards near the sunset, you might spot in your city also. So this whole field is now called active matter in physics, which means they are self-driven out of equilibrium, which means they are consuming energy and moving. It's not like a Brownian motion where thermal energy was making them move randomly. So this is a very big difference. These are out of equilibrium system, like you and I. Human beings are out of equilibrium, and I'm sorry if you are not out of equilibrium, you will be somewhere else, right? So this is what we call active matter, where animals, fish, human beings, bacteria, and also molecular motors in our body. So for example, there are many examples of kinesin motor on a microtubule, the way they move, that is active matter. And what I'll talk today are the artificial active matter, which we make in the lab and make them move as if they have, but because we feed energy. And that is a very, very nice, simple experiment called the vibrating platform. So we have a platform which we move up and down by a very small amount, fraction of a millimeter with some frequency. And the object which is placed on the plate will move in a very directed manner. So that is the active granular matter I will deal with. And the way it moves is, I'll show you how it moves. And recently I thought this connects you and me, because I come from the physics one and you are all the biology people. So there is a very beautiful article few years back in 2016 called The Physics of Life. It's an interesting article to read, though it may claim little more, but very pleasing for physics people. It says from flocking birds to swarming molecules, physicists are seeking to understand active matter and looking for a fundamental theory of the living world. A little bit overstated, but nevertheless, it says that there are many, many things which you can understand from simple principles without going into the details. See, details are very, very important, but a role of a physicist is to see the underlying principles and a minimalistic model to understand the phenomena. And that is what I'll try to show in today's talk. So first attempt to understand flocking came quite a few years back. It's called by Craig Reynolds, and he made a computer model. Actually, you can download that model, is called Boyd, and he just put 3 principles. It said these, whatever you put on the moving thing, they should be repulsing with they should have a repulsion. They should not come and hug each other. It should have attraction also. Some amount of repulsion, some amount of attraction, love and hate, but also alignment. So when this person sees it, he sees what is in the neighborhood and tries to align. So these 3 principles you can put in a simple computer program, and that gave,
it's really interesting,
for
is when you put a
example,
lot
these
together,
inanimate objects.
they behave like a flock of birds or fish or whatever you want.
A collective behavior.
And they do this
What it
with a couple of different
simplified
rules that when combined together in a group, you get this really interesting sort of emergent
if you don't
behavior.
have the right amount,
Like
it
in
does
the
not
buzzword.
happen. And if
You
you
see
put
they
obstacles,
break up
how
and
they
reform
break
and do
and
all
come
these interesting
together,
things
again,
just
very
based on those rules.
fascinating videos you can make. This brought out something very important at that time, because before this, there were very, very crazy theories in very good journals, which were talking of telepathic communication between the birds to make this flock, and so mental templates of orderly flocks. If you read those papers now, it's laughable. but at that time they were in the circulation, and it showed that you really don't need any of this inputs, and you can really have complexity can arise from bottom. This is a very beautiful example of what Aristotle said. So this also was used in Hollywood subsequently for all the animations which were showing the behavior of people running and so on. I'll show you one clip if I get time. It was used, for example, in Batman Return and so on. Many, many movies. It has been used also in Bollywood. So this is something, is the collective obsession where there is an order without command. That is the central theme. There is no central command or director who is telling what to do. They coordinate among themselves to have this collective behavior. And the beauty is now this is what you need in autonomous cars, because you have to take decisions based on what is around you, and flocking algorithms are the ones which are used, and artificial intelligence has many, many flocking swarm algorithms. Even in 1993, there are textbooks on that. So it's a very, very flourishing field. So where did we come in? So when we saw all this phenomena, we thought, let us see how do we mimic them in the laboratory, but with one idea. So I was struck by these videos because this was a part of a paper in Science in 2006. This is what the marching locust video I showed you. And they showed that above a certain number, there is a phase transition, non-equilibrium phase transition from disordered motion to a coherent motion. And this they modeled after what is a very famous model called Vichek model, which means you put all those principles which I told you, but you also put noise in the system, which will disrupt. And it showed that there is a phase where you can get collective motion. But here, the key input is that you would only talk to the nearest neighbor. So this bird talks only to in front, back, left, and right. There is no long range interaction. So our curiosity was, can we have flocking or a collective behavior when the interaction is very long range, not immediate neighbor? This question had not been answered or even asked. So this is what we did. And this was our first paper which came out in 2014. As I said, that's how we started in this game. And Nitin is the one who started for his thesis on this. And subsequently, we have written many more papers, which I will not talk about. So the experiment is very, very simple. What we do, we have a shaker here. Shaker moves up and down the plate, and that plate has this at the top. We put some particles on it here. And these particles, some of them are polar, some of them are not polar, like spherical balls. And we put in a cover slip. And this is a typical particle of a brass. So this is about 4 millimeter long and about 1 millimeter in thickness. Very, very macro object. And what we do, so we shake it at a certain thing. So we are giving energy. So the plate is moving up and down. That is the energy fed. And because of the asymmetry of the particle, it moves. Why will it move? Because when you shake it, this particle will jump up and down. When the particle comes back, there is a coefficient of restitution. Because the particle is asymmetric, it will have a directed motion. As simple as that. It's a very, very simple thing which is known for a long time. But we have used it to make this mimic systems to understand the physics. So this is what we do. For example, this particle which I have shown you, the brass particle, when you are shaking the plate, it will move. And as long as you feed the energy, it will move. But when we change the particle shape little bit, you can see now only one step instead of two steps. You can now see run and tumble. Some of you who are work on bacteria, you might have some resemblance of run and bacteria. And now I can change run and tumble the way I want. I can design a particle which will have the motion which you like. So we can now decide the particle behavior, the active motion, depending on the shape. So I'll give you some examples later on, which we have done further. This is the something new, which is what Professor Rajesh Ganpati has done. I have been collaborating with him. So I showed you only things which move, which means they have a force which is going. Can you have also a particle which not only moves but also has a torque? It can rotate left or right. Moment you bring left and right, you have a chirality. Now you bring the chirality not in shape, but chirality in motion. So the chirality in motion was shown very recently by our experiments. And then you can see that very, very fascinating things arise. I'll come in the end. For example, these are chiral particles. You can see that they are rotating left or right, depending on how we make them. And the other ones are the achiral particles. These are achiral. Now, fascinated by those experiments on locusts, we put these particles on a plate. I have cut down many new other experiments, but let me jump to the flocking. When did I start? I forgot the time. After all the technology hiccups, 4:15. Okay, because I need to keep track of time. Okay, so we put many of the particles, but not enough in number. And what we found that these particles were still behaving not very coherently because the numbers were not enough. So our interest was actually in the long range interaction. So what we wanted to do was bring interaction between these active particles. And so what we have done, we have put the beads, brass beads bought from a cycle shop. These beads are also like few millimeter in diameter. These are like 2 millimeter in diameter. And if I tell you how we procured thousands of them, you will really be amazed. These are not from cycle shop. So we hunted around. We wanted non-iron thing because it should not be magnetic, because this vibrator has a magnet. So what we ended up, and if I tell you the secret after a lot of work by Nitin, we went to Madhya Pradesh and there are people who make rakhis. In a rakhis, you see those beads. Those beads we bought. The only problem was that fellow said, I do not sell anything below 50 because they are so cheap. So I said, no, we don't want 50 kg. We don't even have place. So he said, okay, get lost, take 1 kg free. So 1 kg is still coming handy in my lab. So that is how we converse. So this was because we were doing for the first time. We didn't know how to procure even. But what we found was when we put these beads in between, they still did not have a collective motion. But moment we increase the beads number little bit more, and now you see this is real-time movie, you will see that all these active rods, which we call polar particles, they start moving in a very, very coherent manner. And this goes on as long as you keep on vibrating, because that is the energy source. And this is something which is what we mimicked. And later on, I'll show you, it's a long range interaction because the number of polar particles on the plate are not too many. It's only 6%. And still they form a flocking, not like 50%. So what we showed was that we can now have a transition from with 40% beads, 6% rods, no coherent motion. We can quantify by an order parameter. But when we increase to 5% rods, but 68% beads, you see this beautiful thing. Only 5% rods. That was the most important thing. And then we know what to do as a physicist. You plot a phase diagram with these 2 parameters, how many beads, how many rods. And we found there is a niche area where they form collective behavior, where they do not form collective behavior. And here it's even more interesting, they form jamming and clustering, which I will not talk in today's talk. So this is something we demonstrated. Then we said, let's understand the role of beads, because beads are the eyes and ears for our particles. because we don't have anything else to interact with. When they move in a medium, the beads are displaced. There is a field created there. That disturbance makes the particles interact and behave. And that's what we showed. So what we showed was, when we did not have an ordered state, we looked at the velocity of each bead. Now, how do we do that? We take the videographs. These are every few milliseconds you keep on taking by CCD camera. Then you write the codes. It's quite an involved job. You plot the velocity vector of each bead. You can see there is an arrow here, and they are all randomly placed. But moment you go to an ordered situation, we found that not only the beads, the rods are behaving coherently, even the bead behavior is extremely coherent, which means that the passive particles have become now like active particle by induction. Now, Madan in NCBS, he keeps on telling me there are parallels in biology where the few motors, when they move, can make the difference what happens to something which are surrounding them. And this could have some connection. But what we showed was that beads are also getting ordered. So this is what we showed, for example, snapshot. This rod was very much away from the crowd, but within you can see when they are moving, they are creating a moving rod leads to bead flow, and the bead flow aligns the neighboring rod. And you can see that within 22 seconds, it becomes part of the crowd. And so this is a long range interaction, almost like 10, 12 length away. And still, because of the bead's motion, it comes in its fold. So take home message from this part, which I just thought I'll leave a message here. Collective behavior is seen in many species, which we know. Why is it different with same outcome or same underlying rules? That is the question. Answer is yes. Are the details unimportant? No. The reason could be different. For example, why locusts move in a coherent fashion is very different. For locusts, it is actually the cannibalism, because the locust behind tries to eat in the front person. So this fellow has to save itself. I'm using the right gender, I hope. Then it keeps on avoiding. How he proved it, he took out the gene which feels the pain in a locust. And the moment you take out the pain feeling gene, there was no flocking. Cannibalism was like a survival strategy for the locust. For birds, it can be something else, hydrodynamic advantage. All those things can be different. The details are different, but the very simple behavior, the simple rules can also give you a collective behavior, and it has some merit in understanding. So this is what inspires, and this basic curiosity of in the Craig Reynolds explanation has also given many applications in AI and autonomous cars and so on. Now you can ask, now I'll start asking questions. Can this flocking be disrupted? Because every time flocking is not good. Because supposing you have real life example, flocking in animal groups having a small fraction of baby animals. And it is seen when the baby animal does not obey the order, it actually disrupts the entire elephant flock. There are beautiful videos of that. And a few numbers of baby animals can do this disruption. So it can also be useful in a crowd, because this crowd behavior can be mimicked, which we have done in some other experiments. I will not have time to simulate the stampede. And can you disrupt the crowd behavior by some defects or obstacles to do that? So this has some interesting consequences. So what we did, we took these rods, which I was telling you so far. They form the flocks, which I showed you so far. We call them aligners. But we also have particles. These are again polar particles, of course. But they do not form flocks, even at a very high number. So we call them disruptors or dissenters. So now the question is, how many dissenters you will need to disrupt this? Now that question we are answering experimentally for the first time, because it was known that for static obstacles, they disrupt, but you need a large amount of static disruptors. You need to put many obstacles to do that. What we have shown is now in this experiment that aligners can be now formed flocking even without beads. Please, now I'm changing gears because the numbers are very large, 30%. Earlier, our numbers were only 5%. Their number. We have increased the numbers so that they can talk to each other, and moment you increase their number at 30%, they start forming flocks, which we can quantify by the right appropriate order parameter and so on. What we show now, actually, dissenters even at a 70% will never form a flock. There is no order. Videos are also clear, but when we quantify mathematically, there is no order. Now, when we mix them, and what we have shown, as you can guess, that above a certain fraction, actually they start disrupting and the order is spoiled. So here we have put how many fraction you have to put to disrupt it. 2%, they are still okay. The red ones are the dissenters. But when you increase the number, for example, to 35%, they are disruptive. But they are disruptive even before that. So the mathematical modeling of this can be done. And what you need to plot is the fraction of the disruptors and what is the number of the aligners. First, you get order up to certain fraction, and after that, you will never get a order. So now you can also understand deflocking. So flocking and deflocking are both important. Now the next question comes, can you have two different types of active particles and you can isolate them? For example, if you have two types of bacteria, E. coli and another one which has the different motion, can you separate them? Filter paper will of course kill them, but can you adopt some other strategy? That was the question we asked. And this is what we answered in the following way. Before we answer that, the first step was, how do we trap an active particle? Because unless you have a trap, you will not have a separator. You will not say, okay, one type of bacteria come in this trap, other ones are out of trap. So this we did by again appealing to what bacteria do. So this experiment was known in the literature. Very beautiful experiment in 2003. What they showed was that if you put funnels like this with the appropriate apertures, all these bacteria accumulated only on one side. They do very few bacteria here. This wedge angle is 60 degrees. So we asked ourselves that is 60 degree very sacred or is there a play there? And that is how we started the experiments. And our result was that below 120 degrees, all angles, they trap a particle. When the particles come in, they come in and out, but their fate is sealed and they are eventually trapped. Here when you are close to 120, they come in, but they turn around and go back, turn around and go back. So there is a critical angle which traps them and above which they are never trapped. So that was a good sign. We said, now we know how to design a trap. Once we did that, we know various angles, and this is what is the separation. So there are two types of particles. One are red, one are blue, both are active. And now you see majority of them are red. There is some fellow got stuck by the red, and this is what happens. So you can now separate the particles based on their activity. They are same size. So this is not a mechanical effect. This is an activity trap. So this is the computer simulation, because now we need to understand, because observations are beautiful, but unless you back it up with some nice theory and simulation, you will have a very poor appreciation of the experiments. So what we have shown in the simulation, I will not give you the detail, that two types of particles and only the blue ones are trapped. So what is the difference? I'll give you the reason. What we have done is they're exactly the same size. In one of them, it's a lot of noise, which means the particle is very, very uneasy, keeps on moving, going back and forth. Other one is more persistent. That is the what is difference. And what we are showing is that if you are persistent, you are going to be trapped in life. So only if you are frigidity and active all the time, never let other person predict you, never. Then you will never be trapped. And that's what happens. So this is a trapping because of persistence. Because why? Physically you can see that when the particle comes, get in a trap, poor guy has nothing known, it will keep on pushing the same direction. Whereas if it is a very frigidity particle, it will turn around and come. And that's what happens. So this is what we quantified even in simulations and in theory and showed this is possible. So persistent movers are trapped preferentially, and this is the sorting based on nature of motility characteristic. Now this is one of the recent experiments called non-reciprocal attraction. Non-reciprocal attraction is also a very, very general phenomena of active matter. Human beings, we all know, we are non-reciprocal, right? Because this person likes it, but this person doesn't like. All the time in life, we see that. So this is non-reciprocal attraction. And this happens in nature in many, many species. So the question is, can we understand this non-mutual interaction? And we wanted to do some experiment. So I'll just show you the very heart of the experiment. So what we do now, we take these beads and we put these two particles of the same type. Now what we see is when your internet connection, okay. When these beads are 70%, beads are in a liquid-like state, which means they can move disordered and so on. When we increase their number to 78, it forms a beautiful crystal. You can't see it, but they are arranged in a hexagonal fashion. Beautiful crystal. What it means, this medium does not have any elasticity because it can adjust. This medium has elasticity because they are all very nicely packed. That is how you differentiate between a liquid and a solid. But here, this is a liquid and a solid of the spherical beads. Now, what we showed was that if it is in a liquid medium, two particles, they keep on moving independently and they don't come together. So there is no attraction. But when we put two particles in a crystalline medium, they come together and move in unison. And this is what we used to do experiments in this. So now, in this experiment, what we have done, we have taken a crystalline array of beads. We put two rods, one in this direction moving, one in this direction. So what would you predict? I'll show you the experiment, what it shows, and then give you the explanation. What we see is that immediately this particle catches up with the other one and follows together. In a liquid medium, it doesn't do that. So it means this front particle is oblivious of the presence of this. It is not affected. But the particle which is perpendicular to it really is attracted to the front one and starts behaving cooperatively. And this is the non-reciprocal interaction which I'm talking about. So this is what another video. So the reason behind this, which we have discussed in the paper with modeling and theory, is when the rod moves, it creates a strain field. Now the strain field, because the rod is asymmetric, because it has a head and a tail, the field, the strain decays very, very fast in the front, which means it moves, disturbs the particle, they come to equilibrium very fast. But at the back, what it leaves behind takes a very long distance. So it leaves a trail of destruction. That trail of destruction attracts the other particle because then there is a strain interaction. And that is the physics behind this. So this is what I wanted to summarize, that you can have long range interaction in the between polar particles in an elastic bead medium because of, and then you can get non-reciprocal thing because of this asymmetry in the strain field. Last few slides. 10 minutes, right? Yeah, last few 10 minutes, I'll try to show you now the power of changing the shape as you wish, as you design, and the chirality issues, which are very, very interesting and just beginning to be explored. It's a very, very green field. Very less is known, chiral thing, especially experimentally. So this is the recent paper, not recent, last year's, and I said this is done in Professor Rajesh Ganpati's lab, and Pragya is the student. And what we have done is that there is a emergent stereoselective interaction and self-recognition in polar chiral active ellipsoids. So this extra thing which you will see, that it is stereoselective interaction and it is self-recognizing, which comes out in these experiments. So what is the motivation? We all know chirality, right? Because we know that if you don't have this mirror, this thing, it cannot be superimposed like you in the mirror plane. And this is something which is used in biology all the time because you have too many chiral molecules. And this also gives rise to chiral supramolecular assembly in many systems because of chirality. Now, this chirality is the shape chirality. There is no activity here. Now, this is something known in many examples of supramolecular chemistry, which has been seen. So this being chiral brings about selectivity, self-recognition, sorting, and this is what has been known because of the shape chirality. What we were interested in was in the chiral active matter, where you have not only the internal machinery to generate force, but also to generate torque. And then we can ask whether it is left-handed or right-handed. And this is the design you can make of your particles. So how do we do it? So this is something which people have looked at earlier. For example, instead of the rod which I told you, you can make a, let's say for the pin, which can be bent, and this pin will have the torque, right, when you put it on a vibrating platform. But you don't have too much of freedom here to choose left or right, and this is what motivated us, because in a very earlier paper you showed how the chiral motion happens of this living object also. So what we do here is a designer chiral particles. So we have ellipsoids done by 3D printing. I hope, I'm sure you will have a 3D printer in your lab. You can print 3D particles of any shape or very complicated objects like heart valve and whatnot. But what we are choosing is a very simple thing. These are ellipsoids, few millimeter long and 1 millimeter or 2 millimeter in diameter. But now we make it very rough in one part and very smooth here, which you can do on the computer. You can make the file so that the particle is very rough surface here, and this is a printed particle. It's an actual photo. Now this will move because you don't need asymmetry in shape. You need asymmetry in anything which you want. This will move. But now what we do is, we also bring mass asymmetry. So we make a hole here along this axis. This one will be only the force. It will be like a polar particle. But what you can do now, a clever thing, and this is the work from JNC, they put the hole not on the major axis, but either on left or right. You can immediately see you have the mass asymmetry, which is left or right. And the moment you do that, you can get the chiral particles of different types. How you make the hole, how do you make the particles hollow, all that can be done with different activities, and they will move very differently. You can see that this particle will move in circles which are small radius. This will be slightly bigger, and this one will take a long time. It will move like this. These are actual pictures from the videos which we have taken. And you can see that this is how their particles are moving. They are not only moving with some direction, which is what the force is, but they are also having a torque. And depending on the activity, you can have a choice between force and torque and do these experiments. So we can quantify how they move. That's okay. Now, in the dilute limit, what happens? So what we can do is we can put on the plate the particles which are left moving or which are right moving, depending on how you place them on the table, on the moving plate. This one will tell you how they are moving. So now we can have a mixture which will have plus for me is one chirality, minus is another chirality, left and right. And you can now play with this chi parameter, which means what is the net chirality of the system. So this is a very, very nice system to have play when many things happen. But to cut the long story short, what I'll show you, we take this activity called C2, which has some behavior I showed you. Now you put them together. We put equal numbers here, of course, 1, 1 of left and right. When you put them together separately in the video, they all come together and form a pair. They don't get separated for a long time. And this is a mover. So they annihilate the chirality in some sense and then move like a polar particle. This is what we show, but they also have a spinning motion if you watch it carefully. So they have, some of them form spinners and movers. This is something I will not quantify. So when you put of the same chirality, minus, minus or plus, plus, what we showed is they will always pair up, but beautifully without any glue in between. Because of the chirality and because of the motion, they form the spinners. So C2, C2 of the same type, negative, negative or positive, positive, they form spinners. So you can form spinners when you can form movers. And if you put many of them together, which means, let's say 50 of left, 50 of right, many of them form movers and many of them form spinners. But depending on the chirality, we can show whether they will be mostly spinners or mostly they will be movers. So for some chirality, they will be both possible, spinners and movers. But for the other ones, they only form movers. They never form spinners. It means that the motion is stereoselective. That's what the purpose of this experiment, which is what I have shown. And is there a self-recognition? What we do now, we now put C2 plus minus. You can see we put C4 and C2, this thing. We put many of them together. We ask whether C2 will pair with C2 or C2 will pair with C4 or C4 will pair with C4. All those combinations are possible. These are some examples which to amplify, but the net result we show, which I will again cut short the discussion, that there is a self-recognition that C2 with C2 is the longest lived. It lives almost for 15 seconds. When you have C4 with C4, which is this one, and when you have C2, C4, that is the least one. So we can now show that self-recognition, when you put many of them together, how they pair up. And this is what I meant, stereo-selective as well as stereo recognition. So this is again, I think I'm going out of my time, but what we wanted to also show, again motivated by biology, that if you put these cells, confluent cell layers, it was shown long back, they form beautiful nematic phase, which means they are aligned. and they move in a very nice manner. And there is a recent interest which has picked up that the cell film which you have has a lot of interesting signs. And this is what motivated us to look for in our systems what we can do. So I will probably give only the, if we put many of them together, we can put equal numbers of each or one more, one the other, and the motion is very different. This is equal number, racemic mixture. This is another extreme limit, only pure one. For the same type of particles, you can see the behavior is very, very different. So this is all that has to be understood for the chiral for these systems. And again, motivated by biology, it has been shown that there are topological defects in the cell structures. What I mean by topology, that if you have this nematic particle, they can align in this way or this way. In physics terms, they are called plus half defect and minus half defect. And people have now connected the cancer cells behavior with this defect motion. So there is a very, very exciting literature in the last few years, which is relating the topology defects with the biological action. And that is what motivated us to look for these kind of defects, that we put many of them together and we can show that they form plus defects and minus defect. But in our language, what we want to show, the net result is that for these particles, they never get jammed. They never form a glassy state, which is not the case if they were not active. So the activity helps them never to get jammed. So they will always be mobile. And this is probably the last slide of my talk. So I thought I'll close this to just bring home the point what I started with. Flocking in a small number of rods due to beads mediated by long-range interaction. Then I also discussed that pure active rods in sufficient quantity can also form flock, and you can disrupt this flocking by putting dissenters who do not want to form flocks. And trapping you can do using a wedge below a certain angle, and you can do sorting of active particle based on their activity. Non-mutual attraction, I showed you some nice experiments, and chirality driven chirality active particle showing stereoselective and self-recognition. And the last one was how this topological defects, which are also seen in cell films, they really inhibit the glassy state. So I'll end this talk by the entertainment from this clip, movie called Lion King, which I'm sure you or your kids or grandkids must have seen. You can go to YouTube, and I've taken a small clip from it. And this is done using that algorithm which I told you. So the algorithm of flocking is what is used here. Oh, there is no sound. One second. Actually, if you hear with sound, it's a good effect. Share. Okay, otherwise we'll leave it. Okay. No. Okay, I think you can go to YouTube and watch. But I'll just show you the where this thing has been used. No, it was working when you started.
I can hear it.
Go to the next slide. Okay. Doesn't matter. Just I think we can close this talk. Go to the, just watch without sound. This is 1909 era now. Round is all the
Oh, look, sire. The herd is on the move.
Mufasa,
Okay.
quick! Stampede in the gorge. Simba's down.
So with this, let me thank you, all of you, and I hope you have.
We thank Professor Sood for an absolutely fascinating and insightful lecture, which really took us along on his journey of exploring nature-inspired spines, especially in the domain of self-organized motion. I think you'll all agree he's definitely left us with a lot of food for thought. On that note, we have some time for a brief Q&A session, and we open the floor for questions.
Professor Sood, wonderful talk. I have a quick question regarding your granular flock model. I could see that all the particles were moving towards the edges.
What's that?
Edges towards the as like
yeah,
the centrifugal
okay.
force, as if there's a centrifugal force on the particle. Is it because of the bead sizes or is it because of the
No, no, no. Actually, there is a nice physics that they go to the boundary, and that is exactly what we use to understand the trap. That's what motivated us to make a trap. So what we had done, which some of you would have noted with your keen eye, our edges were in a flower shape.
Yes.
And the flower shape was exactly to avoid the collection of them. And that is told us that there is something about the traps. And that is how we designed the trap. So that led us to the trap experiment.
Okay.
So that was the reason.
Sure.
Correct.
Thank you.
So, Professor Sood, very exciting talk. You know, one of your chiral separation reminds me of the experiments we did in Professor Vijayan's lab in IASC, where we put L and D amino acids together and then do crystallization. So that is where you actually have the angle that is coming in, and you do see head to tail in some, but if you have different sizes of the, chiral entities, they will not form head to tail. So the order that happens is because of the directionality. So that is the comment. The question that I have is, you talked about dissenters for, disturbing the flocks, but the numbers that I saw, if I remember correctly, they were much higher, because you said they should normally be, much less than the personal.
Yeah.
Permanent.
So these dissenters are not so good dissenters. So if we have a dissenter which is very disruptive, then they do it even at 10%. So even at 10%, they started bringing the order down. From 100% order, even at 10%, they had brought, which probably you couldn't read, almost to 60% order. And then 0 became at 35. But there is a very systematic thing, which is what is there in our phase diagram. But depending on how cruel these dissenters are, they disrupt.
Sure. So I think that is, has a lot of validity in today's context as to how many dissenters you should have to disturb any flocking.
Yes. Yes, absolutely.
Hello,
Yeah.
Professor Sood.
Yeah.
Professor Sood, I have a small question.
Yeah.
First of all, thank you very much for such a nice talk. For a vibration, what I meant to say, for the vibration of the plate, is it a regular vibration or what type of thing?
Vertical.
Only
regular.
If you have a irregular,
No horizontal.
No, if you have a irregular type of vibration.
Oh, no. Then it will move, but you will not be able to do much in science.
Yes.
So what we do is the simplest experiment. Now, instead of this, it can also have a complicated shape and all that, which again has its own richness, but we have not explored.
I see.
So, but we are doing, sorry, I forgot to mention we make sure that horizontal acceleration is 0. So we do all the tests And the setup which has been done in JNC, which Rajesh has done, has some very special design to make sure there is no horizontal acceleration. And our experiments are also without any horizontal acceleration.
But if you for the small number of bits and the large number of bits, the large number of bits attains the orderness quickly. If you allow the larger time to the
No, it never.
bits, probably it will.
No. At least in the lifetime of a PhD student, we couldn't do it. I don't know more than that. I mean, I'm sure if you wait infinite times, I don't. But we never got.
Very interesting talk. As you was pointing out, I was also worrying about this circular periphery versus petal-like. And the petal-like one, they were heating and coming out. So I was just thinking of that. When you talked about non-reciprocal attraction, I was thinking about the wording. Is it an attraction or lack of attraction? Or
It's
a lack
call
of
it non-reciprocal interaction.
interaction? Okay. Now, attraction term in any one of this thing probably is not there. It's actually lack of steric hindrance.
Like,
Lack of hindrance.
yeah.
Okay, when you are moving, particles are moving, other things are moving, rods are moving and particles are moving. There's a resistance or hindrance. And when the hindrance is not there, it takes that path. So lack of steric hindrance is a driving force other than the attraction.
No, that's not right. I'm sorry, I couldn't convey correctly. Same thing will be true if you are saying when it was a liquid medium, little below elasticity.
Okay.
Nothing has changed. You have made 75 to 77, all things happen. So only it happens when the medium is responding in a long range interaction fashion. So it is not a steric hindrance at all. So it is the trail it leaves behind, and we have the full.
So by leaving a trail, is it opening space for the next particle to move?
No. Actually, it is 80% packing.
Okay.
There are no spaces.
Okay.
It is a jam-packed crystal. So when this rod is moving, it has to do a lot of work to move through it. So if you don't give enough force, it does not even move. So it is a disturbance which is being created in the medium. Now that medium responds anisotropically. Because it is a head and a tail, head one disturbance dies out in a few 2 beats. Long one lasts for long distance. And when the disturbance is there, the other particle comes and sees some elasticity. So we also showed with the full theory that elastic interaction makes a attraction. So it is a non-reciprocal attraction.
Okay. So is it somewhere slightly related to Bernoulli's concept? No.
Because Bernoulli, sorry, again, I should not brush it aside. Please remember, this is the hallmark of non-equilibrium systems, where we are giving energy, things are moving. Bernoulli principle, which is very important, what we used in our other experiments, is something you don't need non-equilibrium. You have a temperature, you have a pressure, things move, there is a pressure and other term compete, and you get Bernoulli principle. I don't know which Bernoulli, because there is an uncle and a son, and they fought also with each other. So Balu can tell you more on the history of Bernoulli, but the right Bernoulli I'm talking, the famous Bernoulli.
Thank you for excellent talk, sir. Yeah, in lab, you are giving vibrator and you are giving some energy for all these things. But in nature, animal does itself.
Oh, chemical energy.
Chemical energy.
Yeah,
but what is the motive behind that?
Motive of?
Why whole animals comes together and do this?
That's what I said. Probably I said very hurriedly. The reason for flocking, the details could be different. Because this bird which is moving has to sense left, right, front, back, right? Now that is the air disturbance which it is feeling, and that is how they align. But they also get hydrodynamic advantage, because when this fellow moves, this fellow has to spend less energy to follow it. So that's what I said. The reason for flocking in details could be different, but there is a very, very basic principle behind this, which you can really understand from these simple things.
Or birds arrange themselves in the age or efficacy?
In?
Because there are different levels, different age of birds will be flying.
Oh,
together.
yeah. Actually, there is a whole science behind it, and I'm not competent. Actually, when they move in a flocking, the one which is going in the front actually comes at the back after some time, and another one becomes a leader. Then they keep on doing this. Now you will tell they don't have mobile. How do they communicate? But this is the beauty of nature. They coordinate, and this person becomes a leader, then he comes back, and so on. So this is a very, very beautiful thing. And for different species, it could be different. And I told you, human beings are also doing that. Why? Because they don't want to stample. Otherwise, they'll get fall down and get killed. So they have to move. So the reasons could be different in details, but what I'm saying is the collective motion happens in this active matter or the non-equilibrium system, whatever you call it. And that is the basic thing we are trying to analyze. And the key point is the interaction need not be only left, right, back, forth. It can be long distance and still they can feel the presence. That's the beauty.
Hello, sir. Sir at the back. So I thank you for your wonderful talk, and thank you for coming here and gracing us with your presence. I just wanted your opinion on something which might be very abstract, but I found it related. So you are also working on vibrational fields, and there is this school of thought where gravity, they think it is a vibrational field of space-time. So given the increasing disorder that is coming up in our universe, if we look at it, and the galaxies are moving away from each other in the gravitational field of the like vibrational field of space-time. So what is your opinion on this, since given there
sorry, I know your question is exciting, but I am ignorant.
Okay.
No, the reason is whether when the galaxies form clusters and all, there can be many, many reasons, and there can be many ways to do that. I don't think what we see here could be taken and try to see the application. The clustering can happen for various reasons. For example, if I put active particles on a plate, actually they form clumps without any attraction in them. That is called motility-induced aggregation, right?
Yeah.
So, or motility-induced phase transition, whatever you call. That is again a beautiful competition between various forms of reasons. I will not say what entropies and so on, non-equilibrium system, but the reasons could be different. Now, for galaxies, it could be very different. So I will not hazard a guess why it is happening. What I'm saying is that is not the domain I know, and that is not the domain I can even guess, because it could be very different. Yeah.
Sir, here.
Yeah.
Hello. So very interesting talk, sir. Yeah.
Thank you.
So you said you took inspiration from biologists in designing some of the work. So I look at other way around, whether our biology friends can take inspiration from your work.
No,
because
no,
I see, there are, why I'm making this comment.
People are always high. Because they are
No,
always,
no, there are actually, I don't know whether they can think in that line. So one is, there are some disease settings like quorum sensing in bacteria,
yes, yes.
whether they can come
Actually,
up with
it's
some,
very much related.
and other one is cell adhesion and cell migration cancer biology, whether they can use some of these
No,
findings.
I think jokes aside, that's not, I was not meaning that. In fact, in quorum sensing, there is a lot of physics, which really is also studied in physics domains. Actually, that's again a form of collective things,
right?
And it is very much relevant, which I did not touch upon. The second part, cancer biology, I don't know enough at all, but if the way the cancer cells come together and form a bigger things and so on, could have some implication. So I will not know it, but see, the point is, beyond hypothesis, you have to design some experiment which will show this. Then I'm saying it becomes something which one can explore. So that I don't know enough whether there must be experts in the hall, whether that has been done. But I mean, again, I am stepping on the wrong side of my knowledge.
Thank you.
Actually, in the cell last part of my talk, where I said topological defects in a cell film has a lot of implication, that has implication in cancer relation. Because what people have seen is that when you have defects on some things, the defect points really are very active in that area, and that's where cells grow. And that gives the asymmetry. So any asymmetry coming either in the structure, defects are the breakdown of the symmetry, right? That has a role. So people have now seen for let's say some shape where the cancer growth will be predicted and so on. So that's a very fascinating field. And trust me, it's a very, very well posed questions in physics domain now in the last few years. And it's not that we know the answers, but there is a huge sensitivity now to look at them. Huge. Actually, so they are done with real experiments, not mimics. And that's what you're saying is actually has a lot of value.
Sir, I have two quick questions. So the first question is, so you took your inspiration from biology. So did anybody went back to biology and check whether if you change the symmetry of bacteria and then they don't flock?
Oh, that is known because all bacteria do not have this collective motion. That is known. I'm sure your experts are here in the audience. So it's not that all bacteria show this collective motion, which the video I took from the web. No, what you're saying is absolutely right. That is known.
And the other thing is, can I conclude from your beads and rod experiment that it is the long distance interaction is the consequences of the short distance interaction between the beads?
That's correct. No, let me say slowly. When the rods moved in the bead, it created a disturbance. The beads are not in a rigid state. They are quite mobile. They are liquid-like. That communicated, it's like a wind flow. It created a wind. That wind, or I call the motion, whatever is the right word, that affected the particle which was far away and it aligned it. Right? It's the weathercock. Weathercock aligns as soon as the wind direction changes. So it is the wind direction which we are generating in a bead flow.
So is the other way around. The rod motion is changing the bead motion.
Rod motion, first one, is generating the right bead flow, which is attracting a dissenter which is away to say, please come on, join us. And that's what happens.
Thank you.
I'm sure Professor Sood's lecture will continue to inspire a lot more questions, but in the just a
young student.
Oh, one last question then.
Young student, yeah, tell me.
Yeah, so it was related to the last answer that you gave. So when you spoke about the reciprocal interaction, okay, that was happening because the bead concentration was above certain critical point, right? So it went something like a solid kind of.
Absolutely,
yeah.
Absolutely.
So when we, if I try to connect it back to the previous experiment that you have done, where when you increase the bead concentration up till some point, then only you start seeing this flock, right? So ultimately, first thing, can I attribute that interaction to this phenomenon of?
No, I understand. It's a good question. The first part of my talk is when the beads are in a liquid state. So there it is the wind effect, which I was talking. But there the number of beads have to be sufficient, may not be 80%, but let's say 20% or 25%, which really bring the collective motion in the rods.
Yeah.
In the second part, which I said crystal, it is really the elastic strain which is causing even to two particles to come together, which does not happen if it was a liquid, we showed. If it is very few particles, they will never come together. So there is no attraction. So in the crystalline medium only, we talk of attraction. So it is different than the flocking. I hope you understand. Even at 2 particle level, we brought them together. Only 2. It's a very beautiful experiment, even with two particles, but you bring elasticity in the medium. That is the crux of it.
Yeah. Now, when we compare these two to the experiment where you don't have any beads at all, okay, but that requires now a certain amount of this particle to align.
Correct.
Now, that is all. So altogether, if I think it in like if I combine all these experiment, what I am thinking is essential right now is the direct contact between these beads. That is essentially creating this whole phenomenon. So if we don't have any direct contact from the beginning itself, that is not going to help in this flocking.
Correct. So I think it's I understand your flow of thoughts. In the pure rod experiment, the numbers are quite large, more than 50% on the plate. So this rod, when it is moving, it is affecting the, because when you have 50%, the inter-rod distance is less than the rod length. You can calculate, right? Under root of that, 1 over that. 1 over the number square root. That is enough to keep on influencing each other and form this. So that is something, rod-rod interaction, which is kind of coming together, is the reason. Now, when you have beads, these beads, they generate a flow. That flow, it is not that it is always head-on. No, this rod moved and disturbed the beads. That flow, or which you call, what we are calling the flow field, which we can quantify appropriately, makes the alignment. So if you read our papers, we quantify this with the right modeling and physics equations. So we say, okay, you generate a velocity field of the beads. How will it affect the alignment? And above a certain critical number only, this phase transition occurs. So there is a very interesting chain of thoughts which go behind this.
Please join me in thanking Professor Sood for an outstanding lecture. Sir, if we could kindly ask you to remain on the stage. It is now time for the formal release of the CCMB annual report for the academic year 2021 to 2022. This report is put together by our SciComm and Outreach Officer, Dr. Somdatta Karak, and is a formal summary of the research activities and updates from our scientific groups from the past year. It's my absolute pleasure to invite our Acting Director, Dr. Manjula Reddy, to join our esteemed Chief Guest, Professor Sood, on the stage, along with our SciComm Officer, Dr. Somdatta Karak, to formally release the report. So we could ask you to stay back just for one more minute. We are fortunate to have in our midst former directors of CCMB, Dr. Balasubramaniam Durayarajan and Dr. Mohan Rao, whom we now request to join us on the dais to felicitate our honorable chief guest as a token of our utmost gratitude and appreciation.
Where is that?
Thank you to all the dignitaries.
Thank you, sir.
Thanks.
We are now winding down. I'm honored to present the vote of thanks on this memorable occasion, the 35th CCMB Foundation Day celebrations. This year, the event is special, given that it's happening in person after a gap of 2 years. First and foremost, I'd like to thank our Chief Dignitary, Professor Ajay Kumar Sood, for joining us in delivering a memorable Foundation Day lecture. Thank you, sir, for inspiring us with your stimulating science. An event of this scale can only happen with the cooperation of a lot of people from CCMB. I'd like to start by thanking our director for his inaugural address, like I said, all the way from London, reminding us of the history of the institutes, its origins, how and why we came to be, along with an overview of the past year as we celebrate the foundation. I'd also like to thank our acting director, Dr. Manjula Reddy, for her stewardship of this event and for serving as the anchor between all the different groups that came together to organize the events of the day. In that vein, I'd like to thank and acknowledge the members of the organizing committee, especially Dr. Lakshmi Rao, the head of LTS, and the various heads of security, instrumentation, PME, and canteen for their coordinated efforts in putting this event together. Also, a shout out to our student community at CCMB, particularly the organizers and speakers, for their efforts in lining up stimulating talks for us this morning, which started the event off on a high note. Last but not the least, we formally express our gratitude to the director of ISCT, Dr. Srinivas Reddy, for allowing us to use the auditorium for the Foundation Day events and his staff for their help with logistics. All the guests for joining us and making our 35th Foundation Day of CCMB a memorable event. I now kindly invite you to join us for high tea at Shanti Niketan and continue the celebrations. Thank you.